Paula. Today's speakers are Mark Holbrook, Nikunj Patel, and Sebastian Pollack. Mark is Certara's Acting Vice President of Quantitative Systems Toxicology and Safety. He has more than 28 years of pharmaceutical industry experience. Nikunj Patel is a principal scientist at Certara, where he leads oral and dermal absorption projects and is a member of the Cardiac Safety Simulator Development Team. Sebastian Pollack, we call him BASTEC, is a senior principal scientist at Certara, where he leads the development of cardiac safety modeling and simulation systems. Mark, Nikunj, and BASTEC, welcome to today's event. I'll now turn it over to Mark to begin the presentation.
Dan, for the introduction, and it is my pleasure today to introduce to you the concept of Quantitative Systems Toxicology and Safety, or QSTS. I'm going to try to be brief, giving you an introduction and painting for you the vision that we have within Sitara of this exciting area. I'm going to try and leave the lion's share of today's session to BASTEC and Nikunj to take you through some of our exciting data. So, first of all, by way of introduction, what is Quantitative Systems Toxicology and Safety, or QSTS? I'm assuming that many of you may not have previously heard of this term. It's a term we came up with in Sitara following some in-depth discussions that we've had with the biopharmaceutical industry. We recognized that during those discussions, it was clear that there's great variance in the way that in silico modeling and simulation was viewed and was being used by people, and the terms quantitative systems pharmacology, or QSP, quantitative systems toxicology, didn't always capture what people were looking for and could relate to. Hence, we came up with this term in order to describe the scope that we believe is so important. This is a scope that goes beyond toxicology, but also takes into account safety and toleration. So the definition that we've come up with for QSTS is the one as shown on the slide here. This builds on the definition which comes from a recent IQ publication. As you can see, as I bolded, it's a standard description of quantitative systems pharmacology with a focus on drug toxicity and safety and toleration. It is really a key subdiscipline of quantitative systems biology, or modeling informed drug discovery. But when it comes to trying to understand and address safety and toxicology related areas, we need to understand much more the risk associated to organ systems and the relationship of safety to adverse outcome pathways. Now, of course, pharmacology, efficacy, safety, toleration, and toxicity are a continuum. These are all related to the exposure to compound and to metabolite. But the way that we assess these aspects do differ between our different disciplines. When it comes to looking for efficacy, we focus on the primary pharmacology, and we're looking at the pathway associated with that. Whereas when it comes to looking at safety and toxicity, we need to look at an organ system focus. We need to look at both on-target and off-target potential safety issues. We need to look at the chemistry as well. Importantly, whilst every compound may not be efficacious, every compound can be toxic or have safety issues. And this is important by the way we approach this area. So the diagram I've got here tries to paint a picture for you as to how we see these subdisciplines coming together. We don't propose hard boundaries between these different subjects, but more an overlap and an interaction between the different disciplines. At Sitara, we have a strong proven track record in PBPK modeling and simulation with SimStip supporting, as you know, many preclinical and clinical studies and regulatory submissions and labeling. We also use this expertise in order to support our quantitative systems pharmacology approach. And currently, we have successful consortia ongoing focusing in the immuno-oncology and immunogenicity area. For QSTS, our vision is to develop a modular platform, and today we're going to really talk about and focus on the cardiac safety simulator module as a key component of a much bigger vision. So why do we need QSTS? Well, hopefully this slide speaks for itself, and I think it's quite a sobering picture that we're looking at. The current paradigm that we're working to in drug discovery and development is simply not sustainable. Toxicity, safety, toleration issues is costing the industry billions of dollars, and it's hindering the progress of promising medicines through to patients. We're seeing issues when it comes to compound attrition, dose-limiting adverse events, which sometimes restrict the ability to test proof of concept, restrictions in labels, adverse events in the clinic, and importantly, drug withdrawals. And we've been hit by safety issues at all stages in discovery and development. So the figure I'm showing you here on this slide, I think, is quite stark. This comes from the FDA Adverse Event Reporting System and shows a very worrying trend. This is a year-by-year comparison. It runs through to last year, 2017, and as you can clearly see, for all categories, non-serious and serious adverse events and death, we're seeing an increase year on year. Very worrying. Now, the reason for this is not clear, and it may be associated with the reporting behaviors and our ability to capture these adverse events more effectively nowadays. It may also be, of course, be affected by the aging population, the variance in populations, and polypharmacology. But wherever the case, I think you can agree that we need an improved approach to managing drug safety in both the preclinical and the clinical phase. Now, the fact is we have a very big ocean, and if we try to boil that ocean, we're going to fail. We need to concentrate on specific areas. We need to identify a focus. And I've included this slide here, or this table, from a really good literature review, sorry, a very good publication, which comes from Will Redfern and Jean-Pierre Valentin. I don't have time today to really do this work justice, and I'm just going to really focus on the organ system of the day, which is the cardiovascular system. And you can see that this is the top of the league table. It's implicated in the most number of market withdrawals due to safety. And if you look across the row, you can see that drug-induced safety issues with this organ system is an issue at almost every phase, including the non-clinical phase. And this is probably reflective of the high sensitivity of the current herd QT-centric approach of many companies. These data clearly indicate that the current approach is not optimal, and we're going to talk about this in more detail with Bastek and Nikunj. In addition to this, for our QSTS strategy, we've looked at this data, but we've had in-depth discussions and have conducted a survey with the pharmaceutical industry in order to understand priorities, demand, impact, and doability when it comes to what the desire would be when it comes to a QSTS modeling and simulation tool. Not surprisingly, on most people's lists, priority lists, was cardiovascular safety, which was identified as a priority. And this area, I think, is a great introduction for us. It does provide already a good example of QSTS with the SIPR initiative. And here, I think you can see, looking at this diagram, in silico modeling and simulation is playing a central role in this middle-out approach. I like the phrase that, the in silico approach is actually a broker of truth. Here we're seeing it really highlighting the impact of differences in the input data, the difference between different ion channel data sets, such as the influence of assay temperature and static versus dynamic protocols, and then linking this to the clinical data. So from the SIPR initiative, I think we're learning a lot how to develop and then utilize and work as a community to understand how to use modeling and simulation. So we're learning a lot from that. But as you can see from the title to this slide, I really believe we need to build upon this and actually expand our thinking beyond just proarrhythmia. So if you look at this figure on the bottom, this is a pie chart showing market withdrawals due to cardiovascular safety issues. Now, as you can clearly see, proarrhythmia is a major issue. It accounts for nearly a third, but it is second to cardiac injury when it comes to reasons for market withdrawal. Cardiac injury with compounds such as the anorexics, like fenfluramine, for instance, which cause valvulopathy. Cardiovascular events, also significant number of compounds have been withdrawn due to cardiovascular events, such as the non-steroidal anti-inflammatories, the coxib, such as celecoxib, tegaserod, the 5-HT4 agonist for GERD. So the message I'm trying to give you here is that proarrhythmia is key for us. We've done a lot of work to try and understand that, and I think we're learning on how to grow and develop the use of in silico modeling in this space. But we've got a need and an opportunity to broaden from there. So before I hand over to Bastek and Nikunj to take you through some of our experiences with modeling and simulation in the cardiovascular area, I just want to briefly lay out our vision for QSTS. So we believe that this approach has enormous potential. It's interesting and quite disappointing recently, really, that when it comes to the current use of modeling and simulation for the support of drug safety, it's used by a minority of companies. The publication that I'm looking at here comes from the IQ consortium, a survey that they ran quite recently, which showed that only 25% of the companies that they surveyed were actually using such an approach, and that very few were actually submitting their data to the regulatory agencies. The good news is the regulatory agencies are keen to see the data, and they're keen to see the adoption of such an approach. The FDA recently published its predictive toxicology roadmap, and a thing which I see as a great opportunity for us here is the uptake that's already there with PBPK modeling, which has paved the way when it comes to regulatory submissions and the use of modeling and simulation. I like this quote from George Box. Essentially, all models are wrong, but some are useful. Now, the reason I put that quote on the slide is that when it comes to modeling and simulation, I see it as a way to help us move our models forward and to identify the useful ones. There isn't a perfect model. What we can do with modeling and simulation is identify the strengths, the weaknesses, and the gaps. To do this, we really need to work in partnership with subject matter experts in the various disciplines in drug safety, along with the mathematicians, the modeling and simulation experts. We need to bring them together. We need structure in order how to develop such a tool, and again, I think the SIPR initiative has taught us that. The time, effort, energy that's gone into developing SIPR the way it is currently has consumed a lot of energy, and we need to learn from that and figure out how to do it more effectively in other areas. And we also need a focus and a priority in order to develop the useful models. So to close, and before I hand over to Bastek, I just want to show you the vision that we have here within Sitaram. So we see QSTS as a state-of-the-art system that we want to develop. We see it as a modular platform built through partnership with subject matter experts, including the end users, employing our established expertise in mechanistic modeling and simulation with this important philosophy of continuous improvement, i.e., the more we learn, the more we can improve our models. We need to develop and maintain fit-for-purpose models, i.e., there isn't a perfect model. We need to develop them for the right purpose, the right context of use in order to support discovery, clinical development, and regulatory decision-making. And with that, I'm going to hand over now to Bastek to take us through some of our learning with the cardiac safety simulator as what we see as a module as part of QSTS. So Bastek, over to you.
Thank you very much, Mark. Hello, everybody. It's great to run this webinar. Thank you for joining. So I would like to start with some practical issues or problems which we meet every time when we think about the TDP risk prediction or the cardiotoxicity risk prediction. Cardiotoxicity at the level of the electrophysiology of the human heart. So I divided those issues into several groups. I'll start with the first one, which touches upon the source and the quality of the input data which we use for any model, which we use for the TDP risk prediction. It can be in silico model, it can be in cerebral model, or any kind of model. Always the source and the quality of data is absolutely crucial. And here, the SIPA initiative made a huge step forward, because what our colleagues are trying to, or they actually did it, to do is to set up a standard for the measurement of the multiple ionic channels inhibition triggered by the drug. The other problems which we meet are drug-specific, I would say, or at least something what we understand as drug-specific problems. So, for example, which drug is really cardiotoxic, or which drug is really proarrhythmic, or whether the drug which we're thinking about is really proarrhythmic. It's not that easy to define when the drug causes the arrhythmia, or whether the risk is an inherent characteristic of the drug, or depends on multiple factors. So the classification of the TDP risk is of crucial importance. And again, SIPA initiative made a large step forward, at least proposing this classification where we divide drug into three different groups. Then we have to have a clearly defined prediction algorithm and endpoints, so the surrogates of the risk. We know that QT prolongation, for example, is a surrogate of the TDP risk, but it's far, far away from being perfect, so we have to have something to give us the proper information about the risk of the drug of interest. Other issues which can be listed, and which are listed here actually, is the presence of the active metabolite or interacting drugs. So in multiple cases, and most of the drugs are not clear proarrhythmic compounds. Their behavior depends on, for example, the presence of the metabolite or other drugs which are used at the same time. And we know that in a clinical reality, patients in most cases take more than one compound, more than one drug. So the polytherapy is a common behavior, and this is something what we have to consider when we think about the really TDP risk prediction for the drug of interest. There are some examples when the drugs trigger TDP, or at least prolong QT the non-electrophysiological way, which means that some other physiological parameters are disrupted to the level which can cause the TDP, instead of just inhibition of Heric channel. Something what bothers me every time when I see the classification of multiple drugs is lack of the information about the characteristics of the compound, even simple physical characteristic of the compound. It is something what happens from time to time, that we are testing the drugs which are in fact prodrugs, and their half-life and their presence in the body is very, very short. And in fact, what we observed are that, for example, metabolites of these drugs. I mean, instead of testing the prodrugs, we should test the metabolites. There is a large group which I listed here, which is, I would say, patient-specific rather than drug-specific. So the presence of other diseases instead of this one which we are treating at the moment, some physiological, pathophysiological characteristic of the patient of interest, dietary habits, lifestyle, all of them can influence the behavior of the drug. So the question is whether, and if yes, when, we should consider all those parameters, or those which we can describe with the use of our models when we are assessing the drugs for their risk. And last but not least, whenever we think about a model or algorithm which can help us, which should help us to predict the cardiotoxicity of the drug, we have to think about the exposure. So what is the really active concentration, or what would be the surrogate of that active concentration? So instead of using any concentration or any amount of the drug, we should use the proper one. And this is something what I would like to try to address during the presentation. So CIPA in general, at least at the moment, focuses mostly on the early discovery screening, and the algorithms and the models proposed by FDA colleagues and the whole group of the safety pharmacologists involved in this project gives or proposes really good models or algorithms which can help to screen the drugs. But it doesn't end the story because obviously we're not only interested in wiping out the drugs from the pipeline, we're interested in showing when and why the drug can be proarrhythmic. So in fact, it should be a continuum rather than just completely separate model which screens the drug and wipes out those which are potentially proarrhythmic. We should have a continuous model which help us to assess safety depending on the amount of information which we have on different stage of the drug discovery and then development, or even we should take it farther to the post-marketing stage because the number of information, the quality of information increases. Therefore, we can have more impactful insight into the behavior of the drug, which is the drug of interest. Of course, we have to start on the left-hand side. So we have potentially limited amount of information, maybe one channel measured, some information about the exposure. But whenever the farther we go with the drug, the more information we have. So more currents measured, or inhibition of more currents measured, information about the exposure, information about the characteristics of the patients, other drugs which will be taken at the same time with the drug of interest, and so far and so on. So regardless of the place or the level of the cardiac safety assessment, we still can think about in silico models, and they can be useful. That's one thing. And the second thing is we can, at least to some degree, provide information about all those parameters which are listed previously as potential issues. And the tool which we used for all of those models, all of those case studies which we'll be discussing today, is cardiac safety simulator, so the system which allows us to use the different type of information and to predict or to assess different type of surrogates of the proarrhythmic potency. Starting from the left-hand side, as I said previously, and early discovery cardiac risk assessments, the model which we developed used input information about multiple ionic currents and the inhibition of those ionic currents, and the information about the exposure. The important thing is the exposure here was not a real measured exposure. That was the increasing or increased amount of the drug which is present at the place of action. This information can be purely theoretical, or it can come from any source which you can have, like from animal studies or from TPK models. In this case, we just increased the potential concentration of the drug which can meet the channel. And then with use of CSS, we simulated multiple endpoints including QRS, QT, JT-peak, T-peak-T, and an electromechanical window for a population, virtual population of patients. And based or with use of information about 100 compounds or close to 100 compounds which were used as a learning set, and 12 compounds which were completely separated from the learning set and left as a validation, we developed a model which helped us to classify the drug to two groups. potentially prioritic or potentially safe. In this case, our main area of interest was to run this model and to develop this model for the tuberculosis drugs. So all those validation drugs were tuberculosis drugs. But in fact, this model can be used for any other set of compounds, obviously, or any other compounds. But those of you who are interested in the details, they can be found here in the publication, which is given below. But coming back to the main topic of today's presentation, we would like to present the virtual twin concept. So this is not the main area of interest. It's just showing you how we build on increasing number of information. So going further, and focusing on just for a moment on the exposure. So as I said previously, for the early discovery algorithms or classification algorithms or models, we just used a concentration or a amount of the drug in the place of action. But in fact, we know that it won't be a concentration or a amount of a drug. It will be the amount of the drug which is present at the place of action. And at some cases or at some stages of the drug discovery or even development, we don't have a precise information about the concentration of the drug or the active concentration of the drug. With that, what we can do, we can use the PPK model to help us to predict the concentration at the place of action. In general, we tend to use the total plasma concentration as a surrogate of the active concentration. In some cases, it is okay, it is good enough, but as we can see for just one drug, for moxifloxacin, which is presented here, the concentration in the potential place of action, which is heart, and in plasma can be very, very different. Therefore, the question appears, which one is the correct one, and then whether we can use PPK model to predict the concentration. So that was the main question which we asked ourselves, and Nikunj spent really some time on understanding and finding the proper concentration or the proper surrogate of the active concentration. So instead of using just the exposure, we use the PPK model, the developed PPK model. In this case, just for one compound, just for the model compound, which is moxifloxacin, and we tested multiple scenarios. So the scenarios and the question, the scientific question which we asked ourselves was, which concentration, which surrogate is the best predictor for the real active concentration. So we tested 4 scenarios: unbound plasma concentration, total plasma concentration, unbound heart tissue concentration, and total heart tissue concentration. To answer the question, which one gives us the best prediction of the protein potency of this drug? So the endpoint of interest in this case was double delta QT, just because we had access to the information, previously published information about the delta QT, and those 4 concentrations were then in 4 different simulations used to feed the CSS and to predict the double delta. So as you can see, probably the best surrogate was the unbound heart tissue concentration. So those predicted individual values for the double delta QT for the population of individuals, as compared against the observed mean. So those orange dots was the closest when we compare them against each other. The concentration was predicted with the use of the PPK model. The double delta QTCF, so the clinical pharmacodynamic endpoint, was predicted with the use of the CSS, and the information we used as an input apart from the exposure was the information about the inhibition of the ionic currents. Again, the question is, or the question which you may ask is, what is the good quality data when we think about the input information about the ionic channel inhibition? In this case, we used a data or the IC50 value, which was calculated based on the study which was run in the physiological temperature, and which kind of followed most of the, if not all, of the CEPA standard procedure as for the measurement of the ionic current inhibition. So we try to answer the question which of the surrogate will be the best surrogate here. But the question is, and this is the question which I ask every time when I meet my safety pharmacology colleagues, is moxifloxacin really proarrhythmic? In most cases, the answer is no, and I fully agree with that. Moxifloxacin is a perfectly safe drug. So what is the point in predicting double delta QT? In this case, we used it as a model drug only. But what we're really interested in is why and when moxifloxacin can be proarrhythmic. And here is partial answer to this question. So when the moxifloxacin is used in a more or less physiological condition, it is perfectly safe drug. We can expect a prolongation of the QT up to 10, 12 milliseconds maybe, but again, in most cases, that's not dangerous for our patients. But what we have at the bottom of this graph, of the slide, is the situation when we combined not only the inhibition of the currents triggered by moxifloxacin, but we simulated a patient with tachycardia and hypokalemia. So what we can observe is a ECG or pseudo-ECG signal, which is not physiological any longer. I wouldn't dare to say that it's TDP, but it does not behave physiologically. So we have a disruption, clear disruption, what gives us, or what takes us one step farther. So instead of talking only about the moxifloxacin, we're starting to talk about the clinical condition of the patient of interest. So we're not in a discovery any longer. We used all the data which was generated at the discovery stage, but we move forward, adding some information which can come from the clinic, from clinical trials, and we can start to analyze the scenarios when the drug which is potentially safe can be proarrhythmic, or other way around, the drug which we think is proarrhythmic can be really safe. So this is the tool, or it can be the tool, and the first step towards saving the drug or explaining when and why this drug can be proarrhythmic, what gives us the possibility to protect the drug from being withdrawn. As I said, it's just the first step towards the virtual twin concept, and I'll ask Nikunj to take you through the real case study where the virtual twin concept was used. Nikunj?
Yeah, thank you, Bastek. So I think Mark and Bastek gave a very nice background and introduction that leads into the case study that I'm probably going to discuss over the next few minutes. So as Sebastian mentioned, the ability of the drug to block hERG channel or prolong QT is definitely a risk, but the QT prolongation is not the actual risk. The actual risk is the event where there is an arrhythmia induced by the drug. So as Sebastian mentioned, I think moxifloxacin was quite safe in most of the healthy subjects when they were taking only moxifloxacin, but the same drug could actually cause a TDP event or abnormal ECG signal when it was given to a patient with hypokalemia, tachycardia, and potentially an overdose, or maybe a drug-drug interaction with renally impaired patient where the clearance of moxifloxacin is affected. So I think that probably one of the big advantage of such mechanistic modeling would be to identify the clinical safe space, or basically identify the people at risk. So we know that the drug could cause QT prolongation, but actually the simulations could potentially help to identify the patients who can be at high risk of TDP event. And for that, I think we looked into literature. So, can I get the presenter right, Susan?
You should have them, but just give me a sec. There you go.
To Mark's computer. To Mark's computer.
Ah, okay. There you go. Sorry about that.
Okay, thank you.
Yep.
Right, so we picked up citalopram from literature because of various reasons. Because I think citalopram is one of the drugs which is under scanner for cardiotoxicity, and I think there are actually, in fact, 2 regulatory warnings to restrict the dose of citalopram to reduce the cardiotoxic events for citalopram in 2011 and 12. However, there is more to citalopram that basically helped us to pick up this example as example drug for our work. Citalopram has 2 active metabolites that could influence some of the cardiac ion currents. The primary metabolite DCT and the secondary metabolite DDCT. And both of them actually inhibit the hERG channel, and the DDCT is in fact blocks the IKS channel. And there are some studies in literature which basically indicate that the DDCT could be the leading cause of the actual TDP events that are observed after overdose of citalopram. So citalopram, even though it increases the QT interval or it prolongs the QT, it doesn't lead to TDP event at therapeutic doses. I think most of the TDP events are observed after the overdose of citalopram. So I think this made a complex scenario that we could actually try to simulate with the mechanistic models, because we are trying to incorporate drug as well as the 2 active metabolites in the simulations. So to start our work, first of all, we wanted to identify the individual subjects which we can simulate in the computer, because that was our aim. Can we really simulate that cardiotoxic event into a particular patient? So the case 1 was a 46-year female where the life-threatening overdose was taken, about 70 tablets of 20 milligrams citalopram, and the patient was admitted to hospital. We actually identified the cases where the vital parameters, for example, the parameters that are required for the input to the model, like blood pressure, heart rate, and electrolyte concentration, for example, hypokalemia, etc., are measured along with the concentration of drug in the plasma. The case 2 was a 36-year-old female, again overdose of citalopram. Case 3 was a 54-year-old female patient with definitely an overdose, but it was not actually clear how much was the overdose, and it was under the influence of alcohol. And as you can see that the blood pressure and heart rate are quite abnormal with some electrolyte imbalance. So the patient also exhibited hypokalemia. So I think these 3 were the actual cases of the real patients we wanted to simulate, but most of them or all of them were high doses or overdose situations. So we wanted to test the ability of the model to simulate also the therapeutic dose. And at therapeutic dose, I think we relied on the TQT trial results that are available on FDA website for citalopram. So the first question was that what is the exposure level of the parent as well as the metabolites? So I think in most of those clinical cases, the parent concentration or the citalopram concentration in plasma was measured. However, the metabolites are not routinely measured, so we had to calculate the metabolite concentration. One way we could employ is a PBPK model, but at that time we didn't have sufficient data to build a fully detailed PBPK model for parent and metabolite. So we relied on an empirical method to calculate the metabolite concentration. because there is a clinical data showing that the citalopram follows quite a linear elimination even at a high concentration or high dose situation. So that means that the metabolism is probably not saturated at the concentration levels that we were trying to simulate. So with that, we can assume that the metabolite or parent to metabolite ratios are reported in the clinical study at therapeutic dose level. We can apply the same ratios at the high dose situation and can calculate the metabolite concentration. So that was done here. So based on the measured citalopram concentration, we calculated the concentration of the primary and secondary metabolite. And based on the knowledge we had about plasma protein binding of the drug, we could actually calculate the free fraction in the plasma for all the different situations. I think in some cases we had more than one measurement. For example, case 1 was the richest, where we had about 5 different measurements of matching drug concentration and ECG we wanted to simulate. The next question is that once we had good idea about the exposure levels in different individuals, what is the physiology of different individuals? And I think this is routinely not measured because these are quite invasive. For example, the thickness of the ventricular wall, we cannot typically measure it for a live person, as well as the size of the cardiomyocytes, as well as the concentrations of some of the ion channels, etc. So how to fill this gap for a given patient? I think we, based on our historical data of human physiology, we could actually derive some of the covariate models. For example, the cardiomyocyte volume is correlated with the age of the patient. So for example, if we know the age and gender of the patient, we could actually calculate the cardiomyocyte volume. Similarly, we can calculate the ventricular wall thickness if we know the age and the gender of the patient. Similarly, we could actually calculate the heart rate at a given time of the day if we know the age and gender, because there are some prior information available how the heart rate varies with gender, time of the day, and age. So with those type of historical data, we could try to fill the gaps we have in knowledge of cardiac physiology of a given patient, and that was done for this all 4 cases. So there were some parameters that were measured in the clinic, for example, gender, age, heart rate, etc., and the plasma potassium concentration. But some of the parameters like the capacitance of the cardiomyocytes or the ventricular wall thickness, they were not measured in the clinic, and we actually used the covariate models to predict the ventricular wall thickness for a given patient depending on the age and gender. And basically that were fed into the model. So now we know the exposure, we know the cardiac physiology of the patient, so we can actually simulate the QT prolongation or the ECG signal of a given patient under given circumstances. However, the mechanistic modeling could also help you to establish and understand various hypotheses. So if you look at citalopram, there are different literature available giving various hypotheses what is causing the cardiotoxicity of citalopram. So as I mentioned in the beginning, are metabolites important? Is it only the total concentration or the free concentration that is driving the response, or only hERG is important, or we actually need to consider other ion channel activity, for example, IKS or calcium ion channel? So we did various simulations. In one cases, we only accounted for hERG channel inhibition for all the moieties, or we actually looked at only the parent and hERG, or only parent but multiple ion channels. And I think in all those simulations, I think the best prediction of the clinical observation was obtained when we used all moieties. So basically it indicates that the metabolites are important. If you do not account for metabolites when they are affecting the cardiac physiology, I think it could give you a misleading prediction. As well as what it indicates is that you have to account for multiple ion channels. If it is only hERG-centric, I think it could give you a false positives, because there are some other ion channels that could also contribute or actually reduce the effect of hERG inhibition. So this basically shows that when all the moieties were used and multiple ion channel data was incorporated into the simulation, the simulated results were consistently matching the observed QT prolongation for various clinical situations. Now going to the actual simulation of the ECG signal for a given patient. So this is a simulated ECG signal for case 1, or the first case study where the patient had an overdose, but the cardiac electrophysiology was quite normal. So in this case, even though patient had about 70-fold higher exposure than the therapeutic concentration of citalopram, there were no arrhythmias or abnormal rhythms observed in the clinic, or there were no actually TDP. However, the QT prolongation was more than 500 milliseconds, so it was a high risk. But actually, there were no arrhythmias reported in the clinic. As well as in the simulations showed that the QT was prolonged significantly, but there were no abnormal ECG signals. While if you look at the case 3, where the patient also had hypokalemia and abnormal heart rate, again with the same overdose situation, but in this particular patient, in the clinic, they observed ventricular dysfunction with ventricular fibrillation, while even in our simulations as well, we saw that the ECG signal was quite abnormal. So that basically indicates that there is a potential of arrhythmia in this particular patient. So what this case study tried to highlight is that the drug may be carrying a risk of QT prolongation, but this type of mechanistic simulations could actually help to identify the patients at risk, or actually try to help to simulate personalized safety assessment. For example, if this patient is in the clinic or is in the hospital, then basically rather than having a frequent measurements of ECG and plasma concentration, in combination of modeling, I think some of the clinical measurements could be reduced for the patient monitoring in the hospitalization. And with that, I'm going to the conclusions of this talk. So the QSTS approach can really help to reduce, refine, and optimize clinical trials. I think there is a huge potential to run such simulated clinical trials before actually carrying out a clinical study to identify the optimal design that could allow you to establish the objective of the study, for example, the power of the study, etc. Such approach actually help to bridge the translational gap, as Sebastian mentioned, with the moxifloxacin case, so that it allows you to translate from preclinical to the clinical, and actually the simulation of actual abnormal ECG signal simulation, or basically the TDP event. QSTS approach can also have a strong potential for personalized safety assessment, as we showed with the example of citalopram. And again, the QSTS models can actually be adopted in early stages of development, and as more knowledge becomes available about the drug, about the metabolite, about the ion channels it inhibits, and more clinical data, the model can be enriched so that we have more and more confidence built into the model as the drug development program is progressing. And basically, such high confidence models could be helpful to answer some of the clinical questions that are typically asked in the late stage development, or maybe in the post-marketing surveillance signals. And that's the end of the webinar. We are happy to take any questions.
Thank you so much, Nikunj and Mark and Bastek. We would like to now invite our audience to submit their questions for our speakers into the Q&A box, and we'll take as many as we can with the time permitted. So it looks like we have our first question from the audience. When you do ion channel assays, you can do the temperature as ambient or physiological temperature, and you can use a static or dynamic protocol. And these factors can differentially influence the potency of compounds. In the SIPA initiative, this has influenced the ability to categorize compounds as high, intermediate, or low risk. What is the temperature protocol that you've used for the training and test compounds, and does this model accurately categorize the SIPA 28 compounds?
It's Sebastian here. Thank you very much for the question. So for this particular model, we used all physiological, all the data, all the information about the inhibition, which was measured in the physiological temperature. I know it's not in most cases, a high throughput type of measurement. So for the early discovery stage, it would be good to have a model, or it would be desired to have a model which uses the room temperature rather than physiological temperature. In this case, we used all the data which were available in the literature, and we used the best quality data. Temperature was physiological.
Thanks, Bastek. One of our audience members would like to know, we've commented a lot about the utility of the cardiac safety simulator. They'd like to know what are some of the limitations of this approach?
Oh, that's very, very good question. Wide question. As always, there are limitations. It is a model. It is a system which has a limitation as every other system. Just from the top of my head, from the list of potential limitations, whenever we think about physiology and the variability in the physiology, we have to keep in mind that at the moment CSS contains the information about the healthy individuals only. So in fact, it can be not fully realistic, because whenever we go to the clinic, our patients won't be healthy individuals; they will be patients. So they will be, they will have comorbidity, other diseases. They will have diseases at all in general. With that, we are currently working on additional populations, but unfortunately, it's not that easy to find the data. So it just takes quite a lot of time. That's the first thing. The second thing is, just again from the top of my head, the model, the electrophysiological model, or the model of the electrophysiology describes the physiology of the human ventricles, or human ventricular cells. So we don't have atrial cells, for example. With that, the pseudo ECG is not complete, because we don't have a P wave, for example. So that's just one or two examples. At the same time, it is a room for improvement here. So, for example, there is no beta-adrenergic system described in this model. This is something what we're working on. Hopefully, in one of the next versions, we'll have a combined beta-adrenergic system, which will be influencing the electrophysiology. So there are limitations. We're aware of them. They're present everywhere, but still, they don't close the proper and fruitful use of this system. Mark, would you like to add something on to that?
Yeah, thank you, Bastek. And I'd probably just like to add, I'll start generic and then move to more specific. So from a generic concept, we need to develop fit-for-purpose models. We need to move to practical solutions. It's very easy to develop an academic model, something that could be useful to test potential scenarios, but we really want to develop models which are going to be supporting real-world situations. To do that, we really need the engagement of the industry in order to utilize the best input data that we can, understand the limitations of that input data, but also to understand exactly what is wanted. There's a saying, just because you can do it doesn't mean that you should. We could spend a lot of time, refining and developing the cardiac safety simulator, and I think there's room for improvement, no doubt. But maybe this is a call back out to the audience here to get input from you regarding what you would like to see in such a simulator. Maybe that's a comment I'd like to make. Nikun, do you have anything to add?
No, yeah, that's fine.
Yeah. But thank you for the question. It's exactly what we want. So please contact us offline. We'd really keen to hear from you regarding what you're looking for.
Thanks, Mark and Vastek. One of our audience members would like you to comment on what in silico action potential model is used in the cardiac safety simulator, and how does this choice influence the outcome?
I'll try to answer this question if possible. So as you're probably well aware, there are multiple models available in the literature, and the model which is considered as a state-of-the-art model is Haruri model because it's based on its development was based on the human data, and the Haruri model is built into the system and obviously is part of CSS. But as much as I like a Haruri model, and I think it's you know, a really state-of-the-art model. It is perfect for the single-cell type of simulation. So whenever we think about the action potential or modification of the action potential, early after depolarizations, this kind of phenomena, that's probably the model of choice. But at the same time, whenever we go to the more complex simulations, where we have multiple cells connected in the form of a string, I always prefer tensorship model. So all of the simulations, all the results which we presented today were done with use of the tensorship model, the newer version from 2006. It's mathematically more stable. It just simply gives better, maybe not better because it's not correct. It gives the proper information about the pseudo ECG or the pseudo ECG simulated with this model. Is more physiological as compared against the Haruri model. So tensorship model was used. And to answer the second part, or try to answer this second part of this question, whether it will influence the results, probably yes. But we haven't compared that one by one or head to head. We haven't done this kind of a comparison. We did it internally for some other drugs, for some other compounds. Obviously, we run multiple simulations, but we haven't published on that. And whenever we analyzed the data, tensorship model gave us good results, whereas a Haruri model was in some cases mathematically unstable. So that's why we prefer to use tensorship model whenever we run the simulation at the level of the ECG.
Thanks, Vasek. It looks like we have time for one final question from the audience. You mentioned that the quantitative systems toxicology and safety is a modular platform. Can you talk about other coming models and how that will be set up?
Yes, it's Mark here, and I'll try and answer that question. At the moment, we're actually analyzing the results of a survey that we've conducted with the industry in order to understand the priorities, the demand, the demand, the complexity, the doability of the different types of modules that we could look to develop. The types of things on the list include things like seizure liability, kidney function, gastrointestinal function, looking at additional components of cardiovascular safety, such as contractility, looking at vascular injury, such as drug-induced vascular injury. We have yet to decide, and we will be deciding through a consortium that we're considering putting together, which of those areas to focus on first. The ultimate aim, and I'm going to talk big picture now and talk about the future, is to develop, in effect, a virtual patient. That's the ambitious goal that we have. I say virtual patient in quotation marks, going back to one of my earlier comments. We need this to be a practical platform, which has got models or modules which will address key drug safety questions. And each organ system has particular safety questions associated with it that we need to look to see how we can model and simulate. So in summary, we have an ambitious goal. That ambitious goal is, quotation marks, a virtual patient. To get there, we need to focus down on individual modules, which themselves are made up of probably adverse outcome networks or pathways. So we can build mechanistic models. We have, at this point, at the beginnings of a short list, but again, a comment I made earlier, we really want to understand what the industry is looking for. And in order to develop such a capability, we need your input. We need the subject matter expertise that we can partner with the modeling expertise. So sorry, I haven't given you the clearest of answers. I've given you more of a vision there. And I say hit list includes things such as seizure liability, gastrointestinal safety, additional aspects of cardiovascular safety.